the single cell technology really opens up the door to ask new questions and explore the biology of the parasite in ways that were not capable prior.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am Kim Klonowski.
And I am David Peterson, your host for today's episode. Today's guest transforms data on gene expression in single cells into answers to some of the toughest questions in malaria research.
Our guest today is Dr. Anthony Ruberto, post-doctoral scientist in the Center for Tropical and Emerging Global Diseases. Anthony, welcome to the podcast.
Thank you. Thanks for having me.
So, Anthony, one thing we like to do, because we found it really illuminating, is to ask our guests, how did you come to a career in science?
Yeah, so I've always been interested in, answering broad questions, and I've always had this, I guess, sort of intriguing sort of nature of it, just exploring and sort of tackling questions. So I did my undergraduate in upstate New York, and I had a great opportunity at the college that I was at to actually start exploring some questions in terms of really basic biology, in terms of, how fruit flies are able to coordinate their biology. From there, with this experience, I learned that I love being in the lab and sort of exploring these questions and using some of the cutting edge techniques that were available to us. And I transitioned to studying the biology of mammalian hibernators. And this is something that really grabbed my attention. And we're talking about Richardson ground squirrels. So the animals that actually go into torpor during the winter months, and in an adaptation strategy to survive, actually. So, they'll lower their metabolic rate depression for this period of time and then come out of hibernation during the spring to sort of resume their life. And this is really what drove me into biological rhythm research and really understanding the mechanisms that support this ability to sort of lower their metabolic rate, survive, and then resume life when conditions are suitable.
So somehow you got from biological questions with hibernators to malaria research.
Yeah, so if you look at sort of the questions that were being asked with my mammalian hibernator research, it was really on a seasonal or circannual scale, these biological rhythms. From there, I transitioned to understanding, circadian rhythms and how these are important for sort of metabolic health and disease. But interesting with malaria parasites, there are some, if you just look at the blood stages of the parasite's life cycle, these parasites are rhythmic in their own nature as well, where you see them in the blood replicating every 48 hours. So obviously the time scales are a little different, but what grabbed me were these sort of periodic changes that are occurring, from a complex organism that is multicellular to these unicellular organisms that are, capable of sort of sensing their environment and sort of adapting and sort of reading the cues to support their own development. And in our lab, in the Kyle lab, we're interested in one stage of the parasite's life cycle, which is the liver stages. And for one Plasmodium parasite, Plasmodium vivax, they're actually able to enter into this dormancy-like state. And what we're interested in is understanding what are the mechanisms that support this entry into this quiescent state, but also the persistence as well as the exit. And, there's some ties to, again, biological rhythms for these parasites as well.
Well, that's fascinating because I think sometimes students will do undergraduate research asking a very basic biological question, and they wonder how they can kind of move forward in looking at things that are more quote unquote translational. And I think this is a great example of how you can take your knowledge in basic science and find questions, right, that are more biologically relevant in terms of translational potential.
Yeah, I agree. And I think, it's baby steps along the way. And obviously from my undergrad to master's to PhD, I'm learning the techniques that are needed to answer these more complex questions or questions that are indeed more translational. But also I think what's important is just this general sense of curiosity. And I always tell students, you want to do something where you have an interest in the biological question. And from there, things, it doesn't seem like work. You'll go into the lab, you're there for X amount of hours, and it just feels fluid and natural, and you're enjoying your experience. So,
Well, that's a great spirit when it comes to doing research. So I do want to kind of go back to your research. And what can you tell us? You've mentioned the fact that Plasmodium vivax can have this hibernating, if we want to call it, stage of its life cycle called the hypnozoite. Can you tell me what we know currently about what influences the parasite to take on this dormant stage? And maybe what also, I assume you're also interested in how it gets out of the dormant stage.
yeah, absolutely. So at the heart of this, let me just start off by saying that very little is known about these parasites. And that's for a couple reasons. One, unlike some of the other Plasmodium parasites, these parasites aren't amenable to in vitro culture, which means we can't propagate these like we can for some of the other forms of Plasmodium that are available. And then this sense, it sort of limits our ability to probe them and ask questions. Another limitation is the ability to actually study these dormant liver forms. So because it's very difficult to obtain these parasites, it means that we have to go to endemic areas such as, our team collaborates with the Institute Pasteur of Cambodia, where we can source sporozoites. And then we have this fantastic liver stage platform that we've developed in the lab where we can, replicate what it would be like for the sporozoite or the parasite form that is infective coming out of the mosquito to enter into a liver cell, a hepatocyte, and then model this development into, a hypnozoite dormant parasite form.
So is this like an organ culture that you have essentially?
It is essentially a cell monolayer
Okay.
where we source our primary hepatocytes and we put them on a plate, and then with some media we're able to allow the parasites to invade these cells and essentially support their growth. So this is great in terms of drug discovery efforts, but also probing some of the basic biology questions. So this is the model that we've been using typically for studying the hypnozoite. But back to the question of what is actually influencing the development of, the formation of hypnozoites is not really known. So we like to believe that it's a combination of both host factors, but also it being encoded in the parasite itself. So some genetic information that, at the stage prior to entering the liver cell, these parasites are primed to either become a normal replicating form in the liver or persist into a hypnozoite form only to activate months to years later.
So it can even be years later. So they can hang around hiding in the liver and then activate years.
Yeah, so
Wow.
and there's, studies out there that have shown that of individuals that, have moved out of endemic areas and they present with diseases or malaria symptoms, and they'll enter into a hospital and they'll be diagnosed with vivax malaria, which is completely crazy but remarkable at the same time and scary. And actually just about a year ago now, there had been some cases that were diagnosed in Florida and as well as Texas of individuals with vivax malaria. So although the U.S. has eradicated the disease in, the 1950s or so, we still do see these cases. And I do want to say that for these individuals, they had no travel history outside of the U.S. So
That's important to know. Yeah.
what is thought is that perhaps, there are probably small populations or, still residual forms of the parasite that are circulating in the U.S.
Right.
And all it really takes is for someone, military, business person, tourists to go visit an endemic area, get bit, bring their sleeping forms back and have those awake. And of course, we need mosquitoes in the mix.
Yeah, so exactly. So the parasite needs both a mammalian host and a mosquito to continue its life cycle. And for the most part, in the U.S., this mosquito sort of side of the life stage development is pretty much under control. We have mosquitoes, but not to the extent of what we see in some of these more endemic countries.
So I imagine if you don't know very much about the hypnozoite, there's no biomarker to know that someone has a hypnozoite or is infected. So it's just kind of the infection is what tells you, or the clinical disease, I guess, is what tells you.
Exactly. So you could imagine searching for a hypnozoite in a liver is like searching for a needle in a haystack. We have hundreds of billions of hepatocytes or liver cells in our liver. And there's only, again, one or two hepatocytes that could be harboring these quiescent forms.
And I guess it doesn't matter anyway, because if these people hadn't been traveling, he would never be testing for a biomarker anyway. So it kind of is a moot point. But you mentioned something when you were talking about the hypnozoites about targeting them with drugs. So I was wondering if we could move a little bit to talk about drug discovery. Can you talk about whether or not I know you said you know very little about the hypnozoite, but is there something special about this that can be maybe targeted with drugs? And if it's asleep, can it really be targeted? I guess is the secondary question.
Yeah, so some of the hardest forms of parasites to treat are the ones that are metabolically inactive or quiescent. So what we're interested in the lab using our liver stage platform is studying, to see if we can identify new compounds to treat or sort of impact these dormant forms. And this is where single cell biology comes into play. With these newfound technologies, we're able to probe individual hepatocytes that are infected with hypnozoite forms. And from here, using this big data and omics approaches, we're able to identify some of the signatures that are associated with hypnozoite biology. So this is what we're trying to do now. We've probed hypnozoites and we have this awesome data set of transcriptional markers. And with that, we're trying to say, okay, well, if we see gene X being expressed, can we, identify compounds that are targeting the protein of this gene to sort of mitigate sort of the parasites from waking up or actually even entering into this dormancy-like state.
So before we get too much farther into single cell transcriptomics, which is basically a way to measure gene expression in the single cell, can you tell us a bit about the previous technology, which was, I guess, really focused on the whole bulk culture, the whole, a number of cells rather than an individual?
Yeah, so the predecessor to single-cell transcriptomics was bulk RNA-seq or bulk transcriptomics. And this approach was, or still is, very important, but it's limited in is giving this single-cell resolution. So with bulk RNA-seq, you have a signature that is comprised of, I guess, a signature from all cells in a population. So that whether that be a tissue or a cell culture containing hundreds of thousands of cells. And you can imagine that if these parasites, for example, are only infecting one of, 100 million or 100 billion cells, that information from that one cell is going to be drowned in the information of all the other cells that don't contain a parasite. So with single cell transcriptomics, what we're able to do is parse through this population of cells and look at the signatures of the cells one by one, and then any anomaly or any variation from, the population of cells that are not infected, we could then say, okay, well, it looks like from this individual cell, it has a different signature or fingerprint. We also see parasite information coming out of the data set. Perhaps these are the cells that are infected. And how this is important is that you can imagine not only from the parasite side, but the host side, if you're interested in sort of modulating some of the host biology or the cells that are more susceptible or contain the parasites, some of the signatures in these cells may be vastly different than what you see for the non-infected cells. So in terms of drug discovery, to help mitigate off-targets effects or actually be more, specific in terms of some of our therapies, we need to know the signatures of the cells that are infected opposed to the non-infected ones.
I'd never even thought about that part. I guess I was really focused on the parasites and telling the difference between the dormant hypnozoites and the actively developing liver stage parasites. But you're saying that there could be differences in the host cell that's home to these two different parasite types.
Yeah, so with our prior work, what we found was that cells that contain hypnozoites, hepatocytes rather, compared to the non-infected hepatocytes, we do see increased transcript levels of genes associated with energy metabolism, but also we see a decrease in expression levels of genes associated with the host immune response. So whether a parasite are entering these hepatocytes because of these signatures, maybe these hepatocytes are more susceptible to infection, or there are changes that the parasite is inducing on the cell isn't known, but it still tells us that, if you look at a individual infected hepatocyte versus a non-infected one, there are differences. And especially when you're studying these, I guess, rare populations, the single cell technology really opens up the door to really allows us to ask us new questions and explore the biology of the parasite in ways that were not capable prior.
Yeah, this is a really interesting approach, as we've discussed, but mostly because of the fact that you're doing it in a pretty stringently unbiased way, right? So you're not like sorting for cells, for example, that are infected or pulling those cells out. You're looking at it in the whole tissue, but you're able to identify these signatures that you're talking about that you can then use to look at things like whether or not something might be drug targetable. So my question is, can transcriptomics identify a drug target or just genes whose expression is affected by drug treatment?
I could say it could support drug discovery efforts, but I do believe that there are other assays and sort of approaches that are required to validate that. Just because a gene is expressed doesn't mean that at the protein level, if you design a drug that is looking to target the protein of that gene, it is on or accessible or sort of available to target. But I really do feel that it is something that allows, you see, let's look at the parasite's genome. There's over 5,000 genes available. Only a fraction of those are, I guess, amendable to drug therapies. So with single cell or any sort of omics approaches, it allows researchers to hone in more specifically on a subset of genes, and then from here we could parse through it and explore different questions and approaches to actually see okay, well, we see gene X on, what does its protein level look like, to sort of help with this drug discovery.
So I actually want to jump back a bit to the kind of basics on the hypnozoite, because it's really a fascinating stage of the parasite that's in the human infecting malaria. It's unique to vivax, I believe. Is that correct?
There's Plasmodium ovale as well that has been reported to also form these hypnozoite forms. And then if you want to throw Plasmodium cytomolgi in there, obviously these humans aren't their primary mammalian hosts, but they are able to infect humans. And whether, they're able to form hypnozoites is unknown, but in non-human primates, they are capable.
Okay, interesting. But I seem to recall reading that not all isolates of P. vivax have the same dormancy pattern. Can you tell us a little bit about that?
Yeah, so this is where the genetics of the parasite comes back into play. So there are reports where if you have isolates from more northern areas of malaria that have malaria versus more tropical or equatorial malaria parasites, there are ideas that there could be, differences in patency periods and, the ability to form hypnozoites or schizonts. So for example, if you have a parasite that is, say, we have this one strain that's called the North Korean strain. And if we put these parasites in culture, we see that the number of hypnozoites versus a strain that is coming from Brazil, we see that the number of hypnozoites is much higher in terms of these growing forms here. So with that said, it means that we have the same cells that are being infected. It has to come from something on the parasite side that's encoding the ability for them to enter into this dormancy-like state.
And this ties in with transmission seasons, doesn't it, when mosquitoes are present?
Yeah, so there are some hypotheses out there thinking that, okay, well, what is the benefit, or trying to answer the question of what is the benefit of having this prolonged patency period in the liver? And you're right, it could be associated with the transmission season. So you could imagine that, if there's less mammalian hosts around for mosquitoes to bite, that means that perhaps the parasite is somehow detecting, the decreased amount of individuals out there for them to sort of, when they come back out of the human, rather, for the mosquitoes to take them up. So perhaps as an adaptation strategy, they've have this ability now to say, okay, well, we know we're at the end of the transmission season. If we were to come out of the liver and enter the bloodstream, we know that's a dead end because there's no mosquitoes around. It's the end of the transmission season. Perhaps staying in the liver until the next transmission season comes back, this would be beneficial for us so that we could, resume our life cycle into the mosquito. So if that means, persisting for weeks or months, this could make sense. And this all really depends on the environment that you're in. So you have malaria in tropical areas as well as, more temperate areas that have, less rain, but more or so are affected by, I guess, temperature and like dark cycles.
Yeah, it's really fascinating because the time that people have this particular stage is not identical, right? It can vary from person to person.
Yes, and that's true. So on the sort of the host factors that are influencing, the susceptibility, or sort of, let's use susceptibility for them to, host a hypnozoite is not well studied either. So what we know in that is that in the lab we can infect a parasite, a Vivax parasite, into a wide range of various human hepatocytes that are derived from various genetic backgrounds. And we do see this change in the number of hypnozoites or schizons that are developing in vitro. So this, again, tells us that there's something on the parasite side that is genetically encoded, but also there's some influences coming from the host genetic background that is supporting or sort of suppressing the ability for the parasite to generate a hypnozoite.
This is a really complicated problem.
It is, but I think it also speaks to the fact that, we can never look at these things from one side, right? So, I mean, even thinking about like David and I, right? He's usually thinking about the parasite and what the parasite's doing to the host. And I'm usually thinking about what the host is doing to the organism, right? So we always have to look at these things from both sides. They're never one side.
Absolutely. And I do want to highlight that we don't, one of the best things about working and studying or investigating Vivax malaria is the collaborative efforts that are out there. And obviously, as I mentioned earlier, one of the limitations with Vivax malaria is our inability to have these in culture. What that means is that we have awesome individuals in these endemic areas that support us in terms of setting up our ability to have the parasites to use and sort of explore the biology of this stage.
So jumping back to single-cell transcriptomics, you've actually held a kind of a teaching session, a mentoring session recently on giving folks the basics of how to work with the data, because anytime we're talking omics, there's an ocean of data. And the real challenge is, how do you analyze that? How do you sort it? How do you manipulate it? So it's my understanding that session was pretty well attended.
Yeah, no, it was really fun. And I think it was because of the pizza as well, though.
Scientists will work for pizza.
Yeah, so no, it was great. And I had this opportunity with another postdoctoral researcher at the Institute, Dr. Corey Reynolds. And what we did was essentially just give the students a crash course or the opportunity to explore, play with the data themselves. So I guess one of the biggest, hindrances or reasons why individuals don't, play with this data is because could be a steep learning curve. But I think over the course of the 2 days, we sort of to give the students, I guess, the basic tools and some of the confidence to sort of look out there and try to find data sets that, may help with their own research questions, and then sort of explore it using, these pipelines that we've given them.
Well, I can tell you that I can personally thank you for your generosity in holding this workshop, because you've inspired some of my students to actually go and look at some publicly available data sets. So it's been very helpful, and I thank you for your time in doing that. Are there other resources that you could point to for our listeners to assist them in learning maybe in their own area, learning this on their own?
Yeah, so in terms of some of the resources at UGA, there are a ton of courses that are out there through our Bioinformatics Institute as well as the Advanced Computing resource. But just online, I mean, if you want to learn, there are a ton of, I guess, mock data sets that are out there through the Rock Tools suite, but just anywhere. Obviously, if you're a parasitologist, the resources and tools for sort of wrangling the data for parasites is a little less compared to, mammalian systems, data sets that are coming from humans or mice or questions investigating cancer sort of problems. But again, I think just playing with these data sets, getting an understanding of some of the basic steps in sort of pre-processing and filtering things that need to be done before exploring more of the, I'm interested in how gene X changes and condition X versus Y could really be transferred to, any problem. So that's one of the fun things about using single-cell transcriptomics in malaria research is that you have to sort of change the workflows a little bit to make them amendable to the questions for these parasites. So the processing workflows isn't similar to, a data set that would come out of a mouse or a human question.
So before we end, I just wanted to mention, I think that Anthony may be our first postdoctoral fellow on this program.
I think so.
Yeah, so congratulations on that. And also, can you tell us about what's up for the future for you?
Yeah, so it's been a fun time the last couple weeks, obviously looking to transition from postdoc to an investigator myself. So the fall is the time of the year where there's applications and that are out there. So, just interviewing and trying to find a new home to sort of start my own group. So, I mean, my primary questions are, understanding parasite biology and using these various omics tools to better understand these parasites, not only malaria parasites, but any eukaryotic parasites, and then leveraging this information to support, more translational or therapeutic efforts, whether that be drug discovery or, vaccine development approaches.
And we should also mention that Anthony received an award recently as well.
Yes, so I've been fortunate to have received an NIH award, which essentially helps with this transition. So again, I'm fortunate for that, and I think it does kind of help me advance a little bit. So let's see. I mean, with that said, though, with that award, I mean, I'm extremely grateful to obviously the NIH, but also to some of the individuals that have allowed me to explore some of the questions that I present in this grant too. So, and that's both here at UGA with Dennis Cairo in his lab, but also some of my research mentors in France when I was there previously.
Cool. Well, definitely congratulations on the K Award. And with that, our guest today has been Dr. Anthony Ruberto, postdoctoral science in the Center for Tropical and Emerging Global Disease. Anthony, thanks so much for being with us.
Thank you.
Links to additional information about Dr. Ruberto's research can be found on our Instagram account, which is at PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Thanks to the New Media Institute at Grady for use of their Studio Not Found podcast facilities, and a special thanks to our production assistant, Sid Wigand, for research, editing, scheduling, and keeping us on track.